Hello, my name is Molly Langdon and I'm a PhD student at Rothamsted Research. I'd like you to imagine 2050, a date that is currently 30 years away. In 30 years' time, the world's population will have reached 9.7 billion. In 30 years' time, the world will be facing an increase in extreme weather events and drastic changes in climate. In 30 years' time, the world must double global crop production in order to cope with these demands. We, as human beings, are capable of creating solutions to pressing agricultural problems like these. But we only have 30 years to double global crop production, and we cannot wait. We need solutions now. The issue that we are facing is that it can take many years to breed crops to contain beneficial traits. Take wheat, for example. Wheat is a staple crop grown for flour to make bread. 10,000 years ago, wheat looked like this. It had small grains, and the spikes would shatter, sending seed everywhere, making harvesting a nightmare and reducing yields. But modern-day wheat now looks like this. The grains are much bigger and the spikes do not shatter, meaning that more seed can be harvested and subsequently yields have increased. But the problem is that it took 10,000 years for humans to domesticate wheat to what it looks like today. We, on the other hand, only have 30. We are facing additional and immediate challenges of increasing populations and climate change. And we do not have the luxury to wait thousands of years to breed new crop varieties to double global crop production. We need to find new ways of breeding crops to tackle these problems within tens of years and not thousands. There are several methods of speeding up this process, but the one that I work on in my PhD is genetic modification, or GM. So what is GM, and how could it help us? Genetic modification is a valuable tool because it allows us to do things that would otherwise take thousands of years or that would not be possible with conventional breeding. GM does this by creating new genetic variation that does not exist in nature, that we, as humans, can utilise for beneficial purposes. For example, take two varieties of wheat. One with high yields, and another with good resistance to pests and diseases. If you mix these two varieties together, you are mixing tens of thousands of genes. And when you look at the offspring, you may see that some still contain the genes for increased resistance to pests and diseases, but their yields may be reduced because you've lost the genes for high yield. GM, on the other hand, allows you to take just those genes that you require. The ability to be so precise in this manner is what makes GM so valuable. It makes the process of creating crops with beneficial traits so much faster. You are no longer taking two entire genomes and crossing them together, mixing all the genes present, like two galaxies colliding in space. Instead, you are taking one or two specific genes of interest and transferring them across, like plucking a star from one galaxy and placing it into another. And because all life on Earth shares the same genetic code, DNA, GM allows us to look further afield than is possible with conventional breeding. Imagine the situation. You have a variety of wheat that has been destroyed by a certain pest, but current varieties of wheat do not possess the resistance gene for that pest. However, you have found a plant that is unrelated to wheat that does contain that resistance gene for that pest. With GM, you no longer have to worry about trying to get the two crops to mate, which would be impossible for two different species with many unintended genetic effects. Instead, with GM, you can take the specific gene for resistance and transfer it into wheat, quickly and precisely. But just how exactly can you create a GM plant? Genetically modified plants can be created in many different ways, but the method that I use for my PhD is called Transgenesis, and it looks like this. The plant that I am using is called Arabidopsis thaliana, and I am submerging its flowers into a broth of bacteria. The bacteria are called Agrobacterium tumefaciens, and they are a naturally occurring bacteria that transfers the DNA from one species to another. Scientists have utilised Agrobacterium to create transgenic plants. You can engineer the bacteria to carry a specific gene of interest, like your resistance to pests. As the flowers are submerged, the bacteria penetrate into the flower and transfer the DNA into the developing seeds. As the flowers are submerged in a liquid, the whole process has been coined floral dip. Genetic modification has shown promise for solving some critical agricultural problems. From a research perspective, GM has already been utilised to improve responses to biotic stresses and improve crop yield quality in several crop species, including wheat, maize, rice, and soybean. But from an agricultural perspective, GM crops are not commercially grown in Europe. Only one GM crop has ever been approved and grown in Europe. This is because current EU legislations subject GM crops to strict assessment and approval procedures before cultivation is permitted. In addition, half of the countries in the European Union have chosen to ban farmers from growing GM crops by instigating an opt-out clause in the legislation. However, there are scientists and research bodies who are calling for the legislation surrounding GM to be changed. GM will not be the only answer to global food security, but it could be instrumental in addressing some of the challenges that the future of agriculture faces. Although 30 years seems like a long time, 2050 and the need to double global crop production is steadily approaching. We do not have the luxury of thousands of years to breed new crop varieties with increased yields. We need to find new methods of breeding crops now, and GM could be one of those solutions. Time is ticking, and we cannot wait.